So good morning. We continue our discussion related to shuttle. Today we are going to discuss another type of shuttle, glycerol phosphate shuttle. The purpose remains the same. Movement of NADH into mitochondria. NADH, which is generated in the glycolytic sequence, its reducing equivalents must be transferred to the mitochondria where it can donate its electron to the electron transport chain. In that case, we found NADH 2.5 ATPs were generated. Today, we are going to see this different type of a shuttle. Here, instead of 2.5, 1.5 will be generated. We must understand operation of this shuttle to understand this point. This is much more simpler in design. There are only two enzymes, cytosolic glycerol phosphate dehydrogenase, another one by the same name, but it is mitochondria present in the inner mitochondrial membrane. It's an integral membrane protein, E2, embedded in the inner mitochondrial membrane. So in the first step, NADH donates its hydrogens, or more precisely the reducing equivalents, to a compound called dihydroxyacetone phosphate to produce glycerol phosphate. So the reducing equivalents of NADH have entered now glycerol phosphate. And the enzyme is this one, cytosolic enzyme. Now comes the mitochondrial enzyme into picture. The one E2, the one we said is embedded in the inner mitochondrial membrane. The cytoplasmic aspect of this enzyme transfers the reducing equivalent from glycerol phosphate to FAD, resulting in generation of FADH2. And of course, dihydroxyacetone phosphate will be formed. But more important, FADH2 is produced, which is now carrying the reducing equivalents present originally in the NADH. Now this FADH2, as you all know, it can donate its electrons to the electron transport chain to produce 1.5 ATPs. Because the reason is because FADH2 donates the electron to the coenzyme Q, ubiquinol, thereby bypassing the first step of the complex one of the electron transport chain. So anyway, so we have seen that it has got advantage and disadvantage. Advantage is that disadvantage first, instead of 2.5, only 1.5. But the advantage is, as you can see, it is much more simpler in design compared to the earlier one, the one malate aspartate shuttle. Recall, it contained 4 enzyme activity and 2 antipode system. Here, no such complexity is required because the enzyme itself, along with its tightly bound prosthetic group, FAD, is embedded in the inner mitochondrial membrane itself in close proximity to the electron transport chain. I'm emphasizing this term. FAD, it's a tightly bound prosthetic group, tightly bound to the enzyme protein, FA, prosthetic group of the enzyme. All right. So, this way, it is simpler in design. Probably that is the reason that this shuttle is present in almost all the tissues, and that includes brain and skeletal muscles, very important as far as energy requirement is concerned. So now last, before we proceed, you should be able to try to solve this itself. Here it is NADH, FAD को दे रहा है, FADH2 बना रहा है. And what happened in the earlier one, the other type of shuttle, malate aspartate shuttle, there it was NAD and NADH. FAD never came into picture. Now question before we leave, The question is why the FAD-associated enzymes are embedded in the inner mitochondrial membrane, whereas NAD-associated enzymes are present in the mitochondrial matrix. I can give you another example also. In TCA cycle, all the NAD-associated enzymes, isocitrate dehydrogenase, malate dehydrogenase, and what alpha ketoglutarate dehydrogenase, all were present in the mitochondrial matrix. The only enzyme which was associated with FAD, succinate dehydrogenase, was embedded in the inner mitochondrial membrane. What is the reason? Find out yourself. Thank you so much.